두 번째 발표 지금 굉장히 많은 분들이 기다리고 계시는데요. 제가 개인적으로 좀 느낀다면 채팅창에서 뵀을 때 가장 많은 팬을 보유하고 있지 않나라는 생각을 합니다. 지금 이제 대한민국뿐만 아니라 외국에서 지금 우리 이분 두 번째 참가자를 응원하기 위해서 많은 분들이 유튜브에 지금 참가해주고 계시는데요. 그에 힘입어서 제가 영어로 소개를 좀 해드리도록 하겠습니다. 자, next speaker is larissa Miasiro from Chemistry Department. The title is Light Up the Cell. Let's give a big applause. 네, 여러분 큰 박수로 모셔보도록 하겠습니다.
In my lab, we are the magic police. We have an army of ghosts and skeletons. They are tracking different types of magic. They go through the city looking for every house and every room to find the correct magic. So the ghosts, they diffuse through the walls of the houses looking for the magic. When they find the correct one, they hold on very tightly, not to blow away. However, if the houses, they have a protection against the ghost, then we use the skeletons. Skeletons try to go through the doors, but they can only enter the door that has been marked with the magic they are looking for. And once they go inside, they get trapped. Both ghosts and skeletons, they can shine a light very bright when they find the correct magic. It's so bright that you can look when you look through the city. Well, in my lab, we're not actually looking for the city, but actually a human body. So the houses in my story actually represents the cells. So the rooms are actually different types of organelles. The magic we're looking for are the proteins. Each cell type has a different distribution of proteins, and that's why we can use the fluorescent molecules to track them. We have more than 500 different types of cells in our body, so it might be very difficult to find the correct ones. So the fluorescent molecules, they are able to track them and try to pass through the cell membranes and organelles to give us a signal. And they can shine a light very bright once they find their binding partner. They have two mechanisms to do that, like the ghosts and skeletons in my story. So some fluorescent molecules, they can pass through the cell membrane and hold very tightly to the protein they are looking for. We call this hold mechanisms. So they hold the protein as the ghosts hold onto the magic. By the other hand, some fluorescent molecules need to pass through some gates. So like the skeletons in my story, they can only pass the gate if they find the correct partner. When they pass, they get trapped inside of the cell, shining their light. We call this as a gold mechanism. With our tool, we want to light up the cells. So we want to be able to differentiate different types of cells in our body, including cancer cells. And with that, we can map our city and make a healthier and better environment for us all.
자, 발표 잘 들으셨는지요? 지금 채팅창 통해서 정말 많은 분들이 함께 이야기해주고 계시고요. 내용이 어렵지 않아서 좋다. 네, 그 외에 신기하네요. 와, 너무 잘 만들었네요. 라고 정말 많은 분들이 호응을 함께 해주고 계십니다. 자, 우리 심사위원분들은 어떻게 들으셨는지 질의응답 시간 함께 좀 가져보도록 하겠습니다. 네, 우리 이진규 교수님부터 부탁드리겠습니다.
Am I supposed to ask in English?
Please.
Ask questions? Yes. Okay, good. Very good. Thank you. It was very nice. So the molecules, those fluorescent molecules are non-invasive. They do not harm the cell or the organelles and things like that, right?
Yes.
And so we can track down how those molecules, how those proteins work without actually harming the cell.
Yes, that would be the best.
I see.
Yes.
How many molecules actually do you have as candidates for this kind of thing? And are they really, do they really not affect any of the functions of the cell or organelles and things like that?
I see. So my lab is responsible for preparing all these fluorescent molecules, right? Actually, Professor has a library of more than 10,000 compounds. So we have an approach that is called DOFLA. So it's diversity-oriented fluorescent molecules. So what we do is that we prepare different types of molecules and we change their structure of these compounds So we are not actually sure if they're going to bind it or not, but we have a very diverse library. So when we screen that against the cells, then we can actually identify them. So to define if they are toxic or not, in principle, they shouldn't be. So if one of them is toxic, then we can remove them from the list because we don't want them to harm them, right? But there are some specific cases when maybe we would like to use as a treatment. So for cancer, so we found a molecule that if you use in higher concentrations, it might be a little bit toxic, but it's good in this case because we are actually targeting cancer cells. So if you just wanted to visualize, you should be sure that they are not harmful. And if that case, when you find them, you can remove them from the screening and try to use another one that is not harmful. But if your idea is to kill them, then yes, you can use them selectively.
Can I ask one more follow-up question? I'm really curious. Do you have some molecule candidate that you believe to be really safe so they can be used for human embryos and human cells?
We use these in like mice experiments, and they are working pretty fine. The mice are not harmed for anything of that. We can visualize different systems. Yes.
Thank you.
You're welcome.
네, 우리 지금 심사위원분들이 네, 무려 질문을 2개까지 해주실 정도로 많은 관심을 지금 갖고 계십니다. 자, 또 계속해서 혹시 또 질문 있으시면 네, 우리 성민호 박사님, 네.
How did you determine toxicity?
So we have another side experiments that we can do, propidone iodide, or you can also look for the cell morphology. They look dead. You can see that they completely change their structure. So when we are doing the screening and you're applying the dyes, let's say 30 minutes later, you can see that they start shrinking. They say, this is not good. So shrinking means that instead of being like always spread and attached to the plate, they start to make like a circles, like they're making a bubble and they explode. So this is a morphology change. But you can also use some cytotoxic tests to do that. So propidone iodide will penetrate the cells and show you like if it's shining very bright, it means it's toxic. So your cells are already disformed. They open the membrane, so they are dead.
Yeah, so you already have some organ of interest. You're not testing whole body.
Yes, we start in a small scale. So we usually start with the cells separately, already extracted from the tissues. And then you can expand for bigger models, like in the mice, for example.
네, 잘 들었습니다. 우리 네, 이석현님.
Thanks for your interesting talk. It seems like I'm in the kindergarten. By the way, is there any fluorescent probe actually be used in treat cancer or some disease now?
So now we have a lot of probes we already published about, and I think the most recently related with the cancer are Tainier and Taiwai. So actually they can check is a specific type of cancer cells. They are cancer stem cells. So they are even harder to our body because then it can escape from the tumor site and help and go to another part of your body. So they usually are related with metastasis. So we were able to identify only this type of cells with Tainier and Taiwai, 2 different types of dyes. And we discovered that when we apply that at higher concentrations, they kind of accumulate because they bind to certain proteins, and they actually can kill these cells. Slowly, they are slowly toxic. Yeah.
Then, is that kind of fluorescent probe is really used in treating cancer? I mean, not finding in stage of some background research, but really used in...
Yeah, so now we are trying to adapt these probes for PET probes. So usually when you do some kind of tomography tests, you can use heavy metals and use this as a cancer treatment as you actually do in the real life. So we are trying to adapt to that with Taiwai, and now we are preparing different types of Taiwai dyes that contains heavy metals, so we can apply for the common treatments that you use for hydrotherapy. Yeah.
Okay, thank you.
네, 고맙습니다. 너무 잘 들었고요. Larissa, look, they said learning many things, and then your English very good. 당연한 이야기들을 정말 많이 해주셨습니다. 너무 감사드리고요. 네, 다시 한번 감사의 말씀 여러분 박수로 함께 라리사 보내드리도록 하겠습니다. 고맙습니다.